Good morning or good afternoon, and thank you for joining this webinar today. So why am I giving this webinar? I am one of the chemistry directors and the mass spectrometry director at UCSF Health Clinical Laboratories. I started working with mass spectrometry in 2008 when I was a fellow at San Francisco General Hospital, and I have worked with and developed assays, mass spectrometry assays for steroid hormones, therapeutic drug monitoring, and alcohol biomarkers to date. So why do we use mass spectrometry? So as with any methodology, mass spec has some strengths that include potential cost savings when you compare the cost per test to that using an immunoassay analyzer. I would argue that one of the major strengths of mass spec is the ability to separate interferences or other compounds using the liquid chromatography step, enabling the high specificity for your method when you do compare it to most immunoassays where the sample tube is just placed on the immunoassay analyzer. And this can be appreciated by looking at the figure on the bottom of the slide. So on the left-hand side, you can see a comparison between two labs measuring aldosterone by mass spectrometry in patients with chronic kidney disease. And you can see that the correlation is very good. On the right-hand side, you can see a comparison between a mass spec assay and an immunoassay for aldosterone, again in patients with chronic kidney disease. And here you can see that there is a significant positive bias with the immunoassay method, which is leading to falsely elevated aldosterone concentrations in these patients. Another strength of mass spectrometry is that method development can be done in-house and relatively quickly compared to waiting for immunoassay manufacturers to develop the assay, validate the assay, and then of course in the US to get it FDA approved. So some of the clinically relevant analytes that we can measure by mass spectrometry include the steroid hormones, toxicology, including drugs of misuse and pain management testing. We can do therapeutic drug monitoring. We can measure the metanephrines, phenylmalonic acid, homovinilic acid, methylmalonic acid, and also proteins and peptides. So what is the workflow of mass spectrometry analysis? So we first perform some kind of sample preparation where we extract the analyte or analytes of interest from the matrix. We then perform liquid chromatography where we separate our analyte or analytes of interest from any interferences or other compounds that we want to separate. And then we perform mass spectrometry analysis where we have ionization, mass analysis, and detection of our analyte or analytes of interest. And we'll talk about these more as we go through the presentation. In terms of sample preparation, we're not going to focus on that today, but the goal of our sample preparation process is to keep what we want, which of course is our analyte or our analytes of interest. We can either concentrate or dilute them if we need to, and we want to remove as much as possible of the other components of the complex matrix as we can. And so there are a number of different sample preparation techniques that we can use, and these range from a simple dilution or a dilute and shoot protocol all the way up to solid phase extraction, as you can see on this slide. And from dilute and shoot up to solid phase extraction, they get more complex, and you're generally removing more of the matrix as you go up in complexity. And so that's all we're going to cover today in terms of sample preparation. So as you can see from the slide, I'm no artist, but it depicts the basic components of a liquid chromatography mass spectrometry system. And so the sample, whatever type of sample matrix it is extracted first or goes through the sample preparation process and is introduced into the mass spectrometer. And so first of all, the inlet is our liquid chromatography system. We then have in the mass spectrometer our ionization source where the components of the sample are ionized or become charged. We then have some kind of mass sorting by the mass analyzer and detection by the ion detector. And then we have our data analysis where this could be a chromatogram or a mass spectrum. And so first of all, we're going to focus on the liquid chromatography system. And so the basic components of a liquid chromatography system can be seen on this slide. So we have our solvent reservoir, which houses our mobile phases, so usually mobile phase A and mobile phase B. Our mobile phases are degassed using an online degasser. We then have our chromatography pumps, and these produce high pressure so that they pump the mobile phase through the system. We have our autosampler, where our sample is injected onto our chromatography column, and the chromatography column tends to be situated inside a column oven where we can control the temperature. So again, we have our mobile phases and our sample being pushed onto the chromatography column. And then the components of that go through the chromatography column hit the detector. And so in this case, we're talking about mass spectrometry as the detector. And then, of course, we have our computer for our data analysis. So liquid chromatography can be referred to in a number of different ways, some of which are listed here. So LC, HPLC, UPLC, UHPLC. And it's a technique that's used to separate a mixture of analytes in a sample based upon the relative polarity. And that's dependent upon how each analyte interacts between a stationary phase, which is the chromatography column seen in the box here, and the mobile phase, which is the liquid that's flowing through the column. And so there's other interactions at work inside the column, and these include ionic interactions and size exclusion. And so basically the sample, which you've already taken through sample preparation, is injected onto the column in our mobile phase. It's moved along in the flow of mobile phase, which is usually consisting of organic solvent and water. And inside the column, where the stationary phase resides, this determines how long your analyte stays on the column. So how long your analyte stays on the column is dependent upon its interactions with the mobile phase and the stationary phase. So as you can see here, we have a mixture injected onto this column. And then as we go through the chromatography column, we get separation of the red and the blue analyte, where the red analyte is spending more time on the chromatography column than the blue analyte. And so this is how multiple analytes in the same sample, can be separated as they each interact with the column and the mobile phase differently. And so when you're using a powerful technique like mass spectrometry, why do we still need liquid chromatography? And so some of the reasons for this are listed on this slide. So we can use liquid chromatography to separate our analytes of interest from the matrix components or areas of ion suppression. We can use liquid chromatography to separate our analytes of interest from interfering substances. We can use it to separate isomeric or isobaric analytes for mass spectrometry analysis. And some examples are testosterone and epitestosterone, where you want to separate them before they enter the mass spec so you can quantify them independently. And morphine and hydromorphone are also the same, separated in the same way. We can also use the liquid chromatography mobile phases to aid in the ionization process, and we'll talk about ionization in just a minute. And then we can also use retention time from the liquid chromatography system as part of the identification criteria for our analyte of interest. And so what is retention time? So on this slide is an example of a chromatogram. On the x-axis is time in minutes, and this is the retention time of the analyte, which is the time it takes for the specific compound to travel through the chromatography column from injection to detection. On the y-axis is the signal intensity from the mass spectrometer. And so here you can see the retention time is 4.08 minutes, and we have a signal from the mass spectrometer of about 4e to the 5, which is about 400,000 counts per second. So we've covered a little bit about sample preparation and a little bit about liquid chromatography, and now we're going to focus on mass spectrometry. So what is mass spectrometry? So it's a technique that measures the mass of molecules in the gas phase. And in order to accomplish this, charged species or ions are required. Otherwise, there is no way to transfer them through the mass detector. These ions are sorted upon their mass to charge ratio, or the M over Z, and we're going to discuss these things in the next few slides. So in mass spectrometry, our analytes elute from the HPLC or the LC system in mobile phase, and these enter the source of the mass spectrometer. So analyte ions are generated in the source by application of gas, heat, and current by the mass spectrometer. And this is called ion production. These ions then enter the front of the mass spectrometer, and only ions are pulled into the mass spectrometer. All other non-charged molecules are expelled with the exhaust. The ions are then separated based on their mass to charge ratio. Then this is the mass filtering or selection piece of mass spectrometry. The ions are then detected, and so then we have our detection piece, and we'll talk about all of these things in a second. And so what is this mass to charge ratio, or M over Z, that I've been talking about? So the mass of the analyte, M, is the molecular weight when the compound is charged or ionized. And the charge Z is the number of charges on the molecule when it's ionized. And so when a compound is ionized in the source, if we're running the mass spectrometer in positive mode, it will gain protons. And so we get this M plus H ion. If we run the mass spectrometer in negative mode, it loses protons. And so we get this M minus H negative ion. And so if we use testosterone as an example, testosterone can be seen here. So it has a molecular weight of 288.42 grams per mole. If we ionize testosterone in positive mode, it becomes protonated, and so it gains one hydrogen. And so the M is 288 plus 1, which is 289. It only gains one hydrogen, and so the charge, or the Z, is 1. And so the mass to charge ratio is 289 divided by 1, which is 289. So now we're going to go into more detail about the components of a mass spectrometer. So first of all, we'll start with ionization. So ionization is where we convert the flow of liquid from the liquid chromatography system to a gas before mass spectrometry analysis. And as I already mentioned, ions are generated in the source of the mass spectrometer by application of gas, heat, and current. There are different forms of liquid to gas ionization. The most commonly used in mass spectrometry for the analytes that we measure is electrospray ionization, or ESI. We can also use atmospheric pressure chemical ionization, or APCI. And in our laboratory, we currently use ESI for all of our assays. And so you can kind of think about this as converting the flow from the liquid chromatography column as your garden hose to some kind of mist in the ionization source. And so in electrospray ionization, the analyte from our liquid chromatography system is introduced to the ionization source through the spray needle. Solvent droplets, and so this is just a blown up version down at the bottom of the slide. So solvent droplets containing the analyte come out of this charged source probe, and the instrument, again, as I said, can be set to either positive or negative mode, and so we either get positively or negatively charged solvent droplets. In this example, of course, we're using positive mode, and so we get positive droplets. In the space between the end of the needle and the front of the mass spectrometer, we apply gas and heat. And essentially what happens is the solvent is evaporated or driven off. And so this is a process called desolvation. And this forces the charge onto the analyte molecules. And this is the process of ionization. And then one ion per droplet enters the front of the mass spec here. In atmospheric pressure chemical ionization, or APCI, the analyte solution flow from the liquid chromatography column is introduced into this heated tube with help of a nebulizer gas. And this solution undergoes rapid desolvation and vaporization that in turn leaves uncharged solvent and analyte molecules that can be seen in this analyte and solvent vapor. Ionization occurs when the analyte and solvent flow past the corona discharge needle, and this is blown up down here at the bottom of the slide. And so this is where ions are in the gaseous form, and we get ionization. The corona discharge needle is, in this example, positively charged, and so we get positive ions formed. And APCI, or atmospheric pressure chemical ionization, tends to be better for more nonpolar molecules than electrospray ionization, or ESI. And so which type of ionization should you use? Should you use ESI or APCI? And so this table just kind of compares the two. An ESI, or ions, are formed in solution. It has good ionization for polar compounds, and so that means it has good sensitivity for these compounds. ESI is better for thermally labile compounds, and it can produce multiply charged molecules. And this is how we can measure peptides and proteins, even though they have a large molecular weight. It is, however, more sensitive to ion suppression or matrix effects, which can affect your quantitation. Atmospheric pressure chemical ionization has ions formed in the gas phase. So it is good for non-polar compounds. It's also good for compounds containing heteroatoms. For example, if you're measuring the carbamates or benzodiazepines. It is less affected by ion suppression than ESI, and it's more tolerant of high liquid chromatography flow rates. But it does have a limited mass range, and it's not suitable for thermally labile compounds. So essentially, you have to take a look at the analyte or analytes that you want to measure to decide which type of ionization you should use. Okay, so we've covered the ionization piece, and now we're going to talk about the mass analyzer. So we're going to start with a discussion about triple quadruple mass spectrometers. So a triple quadruple mass spectrometer has 3 quadruples, which you won't be surprised to hear. So we have quadruple 1, we have quadruple 2, and we have quadruple 3. So as you can see here, quadruple 2 can also be called a collision cell. So what is a quadruple? It's essentially composed of 4 metal rods, seen here, that are set parallel to each other. And so RF and DC voltages can be applied to the rods of the quadrupole, and this can create an oscillating electric field. And based on the voltages that are applied, it can allow the quadrupole to either act as a filter or an analyte-specific detector for a particular mass-to-charge ratio, or it can just be set to allow all ions to pass through. And so if only an RF voltage is applied to the quadrupole, all ions are allowed through. If RF and DC voltages are applied to the quadrupole, only selected masses are filtered through. And so this can be seen as an example on the right-hand side, where we have the grey ion. It has the mass that we're looking for, and so it is allowed through. But the red ion has a mass that we're not looking for, and so it is lost to the vacuum of the instrument, and we don't measure it. So we can look at this in the context of different scan modes or scan types that we might perform with our triple quadruple mass spectrometer. If we just want to do a full scan or a Q1 scan, in our quadrupole 1, we just scan all mass to charge ratios. So we have our sample, it just goes through, and we scan everything in Q1. So we have it set to RF only. The collision cell is set to RF only also, so we can let all masses through. And in Q3, we set it to RF only too, and it just lets everything from Q1 pass through. So we just see all the masses from Q1. If we're doing a product ion scan, we want to fix the mass to charge ratio on quadrupole 1. So we say, okay, we want this compound and we want product ions of this compound. In the collision cell or Q2, we set it to RF only, so all masses will go through. But we also apply collision gas, and this is to fragment our analyte of interest. And so we'll get to an explanation of that in the next slides. And then in Q3, we want to scan all the mass to charge ratios. So again, we let everything through, and we just see all the masses of the product ions that are formed. And then we have multiple reaction monitoring, or MRM. And this is where we have a fixed mass to charge ratio on quadrupole 1. So again, we tell it exactly which mass that we want to only have ions. All of the ions then enter Q2. All of the ions with that specific mass, enter Q2, where again, we let all ions through. So we apply RF only. And we apply collision gas. So we're fragmenting all of our ions. Then all the ions pass through, and then quadrupole 3, we again have a fixed mass to charge ratio. And so all we're looking for is compounds that have a fixed mass in quadrupole 1 and then a fixed mass in quadrupole 3. And this is called multiple reaction monitoring. And so this is just an example of how the collision cell functions and where fragmentation occurs. So we have our precursor ions from quadrupole 1. So we pick the mass that we want in quadrupole 1, and only ions with that mass will enter quadrupole 2. We then apply gas and heat, as I mentioned, and then we get these fragments or product ions of the precursor ions. And so if we pick the same Q1 mass the same mass in Q1, and then we apply the same gas and heat and voltages in Q2, then this the fragments of the product, fragments of the precursor ions is very reproducible. This will happen in the same way every time if we keep all the conditions the same. And so if we use again testosterone as an example, we have testosterone mass picked as a precursor ion in Q1. So we have our mass to charge ratio of 289 picked in Q1. Everything with a mass of 289.3 from Q1 will enter Q2. Again, we apply collision gas, and then we get our product ions. And so in this example, we have our product ion that has a mass to charge ratio of 97.1, a product ion that has a mass to charge ratio of 109, and of course others. And so testosterone fragments in a very specific way, where we have our testosterone compound here, and here with the lines on the testosterone molecule, you can see how it fragments, and we always end up with our mass to charge ratio fragment of 97.1, one that has a mass of 109, and others. And this fragmentation forms the basis of multiple reaction monitoring. So I explained this a couple of slides ago, but just to go over this again, because this is how we mostly use the mass spectrometers in quantitative data analysis. And so again, we have our testosterone, so it has a molecular weight of 288. When it's ionized, it gains 1 proton, so of course has a mass to charge ratio of 289. So we ionize testosterone, and then quadrupole 1, we pick our precursor ion of a mass to charge ratio of 289. So everything in our sample that has a mass to charge ratio of 289 will go through quadrupole 1 and enter quadrupole 2. So once it's in quadrupole 2, again, we apply our gas and heat and our voltages, and we fragment our testosterone in collision cell 2. In quadrupole 3, we then picked our specific product ions, or the masses of our specific product ions. And so what we're looking for testosterone is a product ion with a mass of 109 and a product ion with a mass of 97, which is what we saw in the previous slide. So this is called a mass transition. So our Q1 mass of 289 and our Q3 mass of 109 is a mass transition, as is our mass in Q1 of 289 and our mass in Q3 of 97. And what we end up with is a chromatogram. And so testosterone, again, flows through the liquid chromatography system in our flow of mobile phase. And this is the retention time. So our retention time is 4.08 minutes. And the intensity, or the counts per second, or the signal in the mass spectrometer is shown on the y-axis. And this is how we perform our analysis in our lab. And so when you're doing multiple reaction monitoring, only selected product ions are detected. And so we have a quantifier and a qualifier ion for each analyte and each internal standard. And so again, we have our chromatogram here. So we have retention time on our x-axis, and we have the intensity, or counts per second, or signal from the mass spec, on our y-axis. And so for testosterone, we monitor 4 transitions, 4 mass transitions, or 4 MRMs, as you can call them. And so you can see here, the chromatogram has different colors. The blue color corresponds to our transition of 289 to 97, and that's our quantifier ion. So that's the ion that we use to quantify testosterone. The red trace is our qualifier ion. So it's the mass transition of 289 to 109. We then have an internal standard. So we're going to talk about internal standards in a little while. But for this example, the internal standard quantifier transition is shown in green, and its mass transition is 292 to 97, and in yellow, which you can't see, the mass of the internal standard qualifier ion is 292 to 109. Okay, so quickly now we're going to discuss a mass spectrometer that has a different type of mass analyzer, and that is time-of-flight mass spectrometry. And so on the top here, you can see a figure depicting time-of-flight mass spec. And so basically, we again have our liquid chromatography system. We have our ionization, where components of the sample become charged. All of our ions are then the same force is then applied to all of our ions, and they travel down the flight tube to the reflectron and back to the detector. And so mass separation in these mass analyzers is based upon the measurement of the time that it takes for an ion to fly this specific path length in the flight tube, so shown by this blue arrow. We can also have quadrupole time of flight mass spectrometry, and it essentially functions in the same way, but we also have quadrupoles, which we just discussed. And because you have quadrupoles, you can select your precursor ion mass in Q1, and you can fragment your either all ions or specific ions in quadrupole 2. And so when we think about time of flight mass spec, it's really important to consider what mass resolution and mass accuracy are. So resolution is the ability of the mass spectrometer to separate one mass from another. And in this example on the left-hand side of the slide, this is what we would see with a triple quadrupole mass spectrometer, so the mass spectrometer that we discussed first today. And so it can distinguish between masses of 868, 869, and 870, for example. And so this has low resolution and decreased mass accuracy. On the right-hand side of the slide, you can see an example for a time of flight mass spectrometer. And in this case, you can see that it can distinguish a mass of 868.50183. And then this one is 869.50580. And as you can see, it can distinguish very easily between these masses, and you can see masses in between. And so this a time of flight mass spectrometer is a high resolution instrument, and it has increased mass accuracy over that of a triple quadrupole mass spectrometer. And so how is this resolution and mass accuracy applied in practice? And so here you can see a cutoff instrument. And if we have two analytes that we want to measure, so in this example, we're trying to measure pentazocine, and morphine. And they both have a mass of 285 on a triple quadrupole mass spec or an LC-MS-MS instrument. But as you can see, using the LC cutoff instrument, the pentazocine has a mass of 285.2093, and morphine has a mass of 285.1365. And so the time of flight mass spectrometer can tell these two compounds apart because of the higher mass accuracy of these instruments. And so time of flight mass spec allows us to unambiguously assign a formula match to an accurate mass measured in a sample, and this facilitates both targeted and non-targeted data acquisition that can allow discovery of unknown or novel compounds. So using TOF instruments, we can perform QTOF instruments, we can perform a full scan of all ions in the sample, and then we can extract the chromatogram to obtain the accurate mass, and then do some kind of database search to identify compounds, as well as matching them based on their retention time. We can also perform a full scan of all ions in the sample, but then we can set criteria such as a certain signal intensity, and this can then trigger the fragmentation in the quadrupoles. And then we can do the same thing, extract the chromatogram to obtain accurate mass and do a database or library search to identify the compound based on the fragmentation pattern, the accurate mass, ion ratios, LC retention time, and we'll get to some of these parameters later. And so we've covered the ionization source and the mass analyzer, and so we're going to talk very briefly about the detector of a mass spectrometer. So the mass spectrometry detector is an electron multiplier. Ions come into the charge plate and are repulsed. The charge horn, shown here, attracts the ions, and this releases electrons as the ions collide with the wall of the horn. The more electrons that hit the wall, the more ions that hit the wall, the more electrons are released, and they're all counted by this blue meter shown here. So when we were talking about multiple reaction monitoring, I mentioned internal standards, and now we're going to discuss what they are. So first of all, we have to talk about some terminology. So a D equals deuterium, and it's a heavy isotope of hydrogen. And so here you can see hydrogen has a molecular weight of 1, and it has a proton in the nucleus. On the right-hand side here, we can see deuterium. It has a molecular weight of 2 because it has a proton and a neutron in the nucleus. And so if you see an internal standard written like this, D with an X, this means that deuterium has been substituted for hydrogen in a molecule to change its molecular weight. And so an example would be cocaine. So we have cocaine, we have a molecular weight of 303, and then we have D3 cocaine, which has 3 deuterium here in place of 3 hydrogen. And the molecular weight of D3 cocaine is 306, so it's 3 mass units heavier than cocaine. And the same can be applied to carbon-13. So carbon-13 is the heavy isotope of carbon. So carbon-12 has 6 protons and 6 neutrons in the nucleus with a molecular weight of 12. And carbon-13 has 6 protons and 7 neutrons in the nucleus and has a molecular weight of 13. So when we're thinking about internal standards, if you see 13C with an X, this means that carbon-13 has been substituted for carbon-12 in a molecule to change its molecular weight. And so if we use my favorite testosterone as an example, as we've already discussed, the testosterone mass to charge ratio is 289. If we have 13C3 testosterone, so there's 3 carbon-13s, on the testosterone, the molecular weight is 292. And you can see here the stars are showing where the carbon-13 has been substituted. So what is an internal standard? So we add an internal standard in the same amount to all of our samples when we're doing our batch analysis for mass spec. So this is added to our calibrators, our quality control material, and any matrix-based samples that we're running. We most often use deuterated internal standards, and that's mostly because they are more frequently available, but carbon-13 internal standards are becoming more available and are becoming more cost-friendly. So the deuterated or carbon-13 labeled internal standard has the same chromatographic properties as the non-labeled compound and flows through the LCE or the liquid chromatography column with the analyte, and so it has the same retention time. But the mass spectrometer can tell them apart, right? So the internal standard is heavier than the analyte of interest. And because of this difference in molecular weight, the mass spectrometer can tell them apart. And so we add the internal standard to every sample. And ideally, it has the same retention time as the analyte, but it has a different mass. And so you've seen this example before for testosterone. We have our retention time in minutes. And we have our 4 MRMs or transitions for testosterone. And as you can see with the green trace, it has the same retention time as the blue and red traces for testosterone. So the green trace is the testosterone internal standard. This is a D3 internal standard, so it has 3 deuterium. And so its Q1 mass is 292, whereas testosterone has a Q1 mass of 289. So of course the mass spectrometer can tell them apart. But they behave in the same way. The internal standard behaves in the same way chromatographically as the analyte. And again, the testosterone breaks apart. You've seen this before, the fragmentation of testosterone into 97 and 109. And you can see the same for the internal standard, where we have our deuterium up here, where it has a mass to charge ratio of 292, but it also forms the fragments with 97 and 109. And so why do we use internal standards? Again, we add it in the same amount to each calibrator, sample, quality control material, and we add it as soon as possible, so usually as the first step in any sample preparation process. And the reason that we do this is the internal standard can help adjust for variance in the sample extraction procedure or the sample preparation procedure, variance in the liquid chromatography injection volume, and then variance in the mass spec response. So this can be in the ionization process, the transfer of the ions from the source into the mass analyzer, the mass analysis itself, and the detection. And it does this, the adjustment takes place because when we are doing quantification in mass spectrometry, we perform it using peak area ratios. Now, what do I mean by that? So the peak area ratio is the analyte peak area divided by the internal standard peak area. And so because we add the internal standard in the same amount to every sample that we're analyzing, this essentially normalizes or adjusts for things that can happen during the extraction process, the LC process, or the mass analysis process. And so what we do with the internal standard peak areas is monitor them during a run as an acceptability metric to detect possible issues with our sample preparation, our injection, or our instrument, etc. Okay, so now we're going to move on to discuss some of the important aspects of data analysis that we do when we're doing mass spectrometry. And so when we think about quantitative data analysis, so when we're trying to quantify our analyte of interest, there are a number of things that we can look at. So these include our analyte and internal standard peak review, the retention time, peak integration or peak shape, our signal to noise ratio, our calibration curve, our quality control results, our internal standard response and recovery, if we see any carryover or contamination in our system, ion ratio monitoring, and more. And so for the purposes of this presentation, we're going to just look at some of these. And so I mentioned before that the retention time of your analyte is the time it takes from the injection onto the LC system until it's detected by the mass spectrometer. And so in this example for 25-hydroxyvitamin D3, the retention time here is 1.94 minutes on the left-hand side, of the slide. On the right-hand side of the slide, we can see the peak for the internal standard for 25-hydroxyvitamin D3, and it also has a retention time of 1.94 minutes. So the absolute retention time of 25-hydroxyvitamin D3 is 1.94 minutes. But what we can also do is calculate the relative retention time. So we can take the retention time of the analyte and divide it by the retention time of the internal standard, and that would give us the relative retention time. And in this case, because they're the same, the relative retention time is 1. And so either the absolute retention time or the relative retention time or both can be used as acceptability metrics for your mass spec run to make sure that you're identifying the correct peak and to make sure that your analyte is not shifting in relation to your internal standard. Another thing that we tend to look at during our mass spectrometry analysis is our peak shape. So do we have acceptable peak shape? So the figure here on the left-hand side shows a testosterone peak that has good peak shape. In the middle, we can see the testosterone peak that has a shoulder here on the right-hand side of the peak. And then on the right-hand side here, we can see that this testosterone peak has fronting on the front of it. And so if you're seeing unacceptable peak shapes, such as shouldering or fronting, then there's a number of possible reasons that could cause this. And so if you are seeing tailing, it could be the incorrect gradient, so the liquid chromatography system has some kind of issue. It could also be that you potentially have an underlying interfering peak. So when we're doing our method development and our method validation for mass spec assays, we often, look at interferences that we know could cause a problem with our assays. So, if you think about testosterone, and epitestosterone, you want to make sure you separate them before they enter the front of the mass spectrometer. So separate them on the LC system. But there's going to be things that you don't know that might cause an interference. And so if you are seeing some kind of tailing or fronting, it could potentially be an interfering peak. If it's a new thing that you haven't seen before in another sample. If you're seeing fronting on your peaks, it could be that the concentration of your analyte is too high that you're injecting, and so the capacity of your chromatography column has been exceeded. But again, it could also be an interfering peak at the front of your testosterone peak, for example. In general, the peak shape could just be some kind of strange peak shape, could be some kind of interference. And it could also be that maybe your peak is very, very small. And so you might just have high background, or you might have a very, very small peak. And so the signal to noise ratio is potentially low. So what is a signal to noise ratio? So if we take a look at this chromatogram here, this is 10 nanograms per deciliter of testosterone. And we have the signal, which is essentially the peak. And then we have noise, right, which is here on the baseline. And so in the software for most mass spectrometers, you can tell the mass spec what is noise and what is signal, and then it can run a calculation in the background. And so in this case, the signal to noise ratio that we have here is 136 to 1. And so recommended minimums for signal to noise ratio vary for accurate quantification, but in general, it's thought that a signal to noise ratio of about 20 to 1 is good for accurate quantification. And so when we're thinking about developing our method, how do we determine if what the signal to noise ratio is? So you want to know this, you want to find this out during method development to make sure that you have sufficient signal for the analyte that you're trying to quantify. And so one way to do this is to spike your analyte of interest. So again, we're using testosterone as an example here. Spike testosterone into different lots of stripped serum. So this is charcoal stripped serum that removes the testosterone out of it. This was done with 8 independent extractions and was analysed over 2 different mass spectrometry runs. And so if we put the data together, we can see that the target concentration for testosterone was 1 nanogram per deciliter, 2.5 nanograms per deciliter, and 5 nanograms per deciliter in these different samples. The mean concentration that we actually achieved is seen here. And then at the far right-hand side, you could see the signal to noise that we achieved based on these concentrations. And so here we can see our 1 nanogram per deciliter example. Here we can see our 2.5 nanogram per deciliter example. And here we can see our 5 nanogram per deciliter sample. And so you can see the signal is very small at 1 nanogram per deciliter. It gets bigger at 2.5 and then gets bigger again at 5. But depending on your criteria, so if you're using a 20-to-1 signal to noise ratio as your acceptability criteria, you would not be able to accept any of these concentrations as your lowest signal to noise. And so it may require you to go back and re-optimize the mass spec parameters for your compound, or potentially your sample prep or your LC, in order to try and increase the signal to noise. And so say that you developed your method, but you don't have acceptable signal to noise. It could be that your analyte concentration is just too low for valid quantification. It could also be that potentially you have ion suppression or matrix effects. So one of the reasons we do sample preparation is to try and minimize our matrix effects, or minimize the ion suppression. So in the ionization source of your mass spectrometer, the analyte that you're trying to quantify is competing for charge with all of the other matrix components. And so if there's a lot of different matrix components in there, there's going to be a lot of them getting the charge that should be on your analyte. And so you won't get as high a signal for your analyte. And so again, you could potentially re-optimize your sample preparation process if this is causing a problem. You can also optimize your liquid chromatography system to sort of move your analyte out of areas where there's a lot of background signal or matrix effects or something eluting from your chromatography column. If you don't have acceptable signal to noise, again, there could be a problem with your sample preparation process, and it could be that you have high background, as you can see in this example. And sometimes that's something that occurs over time with your assay, so you don't see that in the beginning, but potentially, once you've been using the assay for a while, the chromatography column is potentially getting old, or if it's become contaminated with matrix components, you start to see higher background, and it could be time to switch your chromatography column or to clean your mass spectrometer. So how do we actually quantitate our analytes of interest? And so luckily mass specs come with software that helps us to do the data analysis. And so on this slide, you can see a table at the top that has the sample name. And you can see on the left-hand side here, this is the analyte peak. On the right-hand side here is the internal standard peak. And then we can calculate the area ratio, which I described before. So we take the analyte peak area and divide it by the internal standard peak area. And then this peak area ratio is used to construct a calibration curve. So as I said, in our batch of samples, we usually have calibrators, our quality control materials, and then our matrix-based samples. And so for the calibrators, we know the concentration for each calibrator level, and so we plot this on our x-axis. And then on the y-axis, we have the peak area ratio, which is the analyte peak area divided by the internal standard peak area. And so we can construct this calibration curve based on the known analyte concentration for each calibrator and the peak area ratio that we obtain. And then for our unknowns, for our quality control materials and for our matrix-based samples, we then calculate the peak area ratio for them, and we can use this calibration curve to then determine the concentration of the analyte within each of those samples. So as I mentioned, another parameter that we should monitor is the internal standard recovery within a batch of samples. And so basically here on the right-hand side, you can see this is the injection number along the x-axis, and you can see the internal standard peak area is fairly consistent across the whole batch of samples here. And so what we can do is calculate the average internal standard peak area of the calibrators within each batch or within each run. And then we also, of course, have the internal standard peak area of each sample within each run, and that's what can be seen here. And if you want to set acceptability criteria for our quality control materials or our matrix-based samples, an example of this would be that the internal standard peak area of each of these samples within the run has to be within plus or minus 20% of the average calibrator internal standard peak area. That's just an example of criteria that you can use to accept the internal standard response in each run. If, of course, your internal standard for a particular sample is outwith of that criteria, then you potentially might need to do a re-extraction, a rerun of that sample. If you think that potentially it was misinjected, for example, you could try re-injecting the sample to see if the internal standard peak area normalizes. Another thing that we use frequently in our mass spec analysis are ion ratios. And so on this slide, again, you can see a chromatogram where we have retention time along the x-axis and intensity or counts per second on the y-axis. And here we have testosterone, our favorite example. So the blue trace here is testosterone transition 1, or the quantifier transition, so 289 to 97. And in the red trace, we have testosterone transition 2, or 289 to 109. And what we can do is calculate the ion ratio for these. And so for our ion ratio, we take the peak area of the 289 to 109 transition and divide it by the peak area of the 289 to 97 transition. And in this case, we get 0.96. And then what we can do is use our ion ratios to identify excessive background in our run, to identify peak asymmetry or abnormal peak shape, or potentially identify the presence of interfering peaks. And so these ion ratios are very consistent across all of your samples within a batch. So you can calculate the ion ratio for every single sample, your calibrators, your quality control material, and your matrix-based samples. And again, you can use the calibrators to set the criteria. So that an example acceptance criteria would be that the ion ratios for the quality control and matrix-based samples should be within plus or minus 20% of the average ion ratio calculated from all of the calibrators within a batch. And one of the reasons that we do this is seen on this slide. And so again, we have our testosterone. So in this example, we have a calibrator. So we have our 289 to 109 transition peak and our 289 to 97 transition peak. And here, the ion ratio is 1. In this example, from a matrix-based sample, we have our 289 to 109 transition and our 289 to 97 transition. But in this case, the unknown sample ion ratio was 1.45, which is out with our 20% criteria. And you can kind of see here, but on this peak, we can see that there's a hump. And so this can be indicative of an interference, in which case you wouldn't want to report the sample result, and you would want to potentially send it to a different lab for analysis. So we've been talking about quantitative data analysis, mostly using our triple quadruple mass spectrometers, but you can also perform qualitative analysis if we're using our time of flight mass spec. And so we have our sample preparation, as we discussed earlier, and we have our liquid chromatography, and we have our time of flight mass spectrometry analysis, and then we get our chromatogram here. And so what you can do is extract the chromatogram, and you end up with our retention time, the accurate mass for our compounds, and then we can do this database search. And so we can search our compound database to see if our retention time and accurate mass matches any of the compounds in the database. And then the software will actually assign a target score for each compound in the database based off of these two criteria. It will also calculate a mass error. And so you can get an idea of how sure the software is that your compound is actually present in the sample. And so it's essentially is the compound there, is it not there? So our qualitative analysis. For each compound in the list, you can actually also take a look to see how well the mass spectral library matches what is seen in the patient sample. And so if we look here at methamphetamine, the third compound in the list, you can see that the target score is 98.3. And the software has another score, 98.89 for the target score. And then it has, the software has another score that's the MS library matching score of 98.3. And so then you can see this is the mass spectrum of the actual sample, and this is the reference standard for methamphetamine that was used for building the library. And as you can see, these are a mass spectral match. If we look at ketamine, which is the second compound down, we can see that it has a target score of 85.5, but it has no score for the MS library matching. So if we take a closer look at the ketamine retention time, then this is the sample MS spectra for the species that's there. And this is the ketamine reference standard from the library. And as you can see, this is no match. And so this shows how you can increase the selectivity of your method by using the QTOF to add the MS library matching score. And with that, I would like to acknowledge a few people. The MSACL LCMS 101 short course content, some of it that are used here, it was developed by Grace van der Gugten, Judy Stone, Laurie Bachman, Jackie Hubbard, and Grace Williams, and I'd like to thank them for their help with that. The QTOF and TOF data came from Dr. Roy Girona, and so I'd like to thank him for that. I would also like to make a shameless plug for the MSACL conference. It's in October of this year. It's a wonderful conference. It's very heavily mass spec orientated, and it's one of my favorite conferences. You can scan this QR code and it will take you to some more information about it. I am very lucky to be one of the instructors for the LCMS 101 short course at MSACL, and so it's getting started with quantitative LC-MS in the diagnostic laboratory, and you can see some more information about the short course here or again on the MSACL website. And my friend and colleague Julie had this problem-solving flowchart in one of her mass spec presentations, and I thought it was very applicable to mass spec, and I thought it was funny, and so I thought I would include it here too.